Hello everyone, welcome to the channel Microbial World. Today let's learn about conjugation in bacteria. Conjugation is a process where bacteria can transfer genetic material from one bacterium to another. The process involves direct transfer of DNA from donor bacterium to recipient bacterium. DNA is transferred in the form of plasmid. So what is plasmid? Plasmid is a small circular double-stranded DNA molecule that is separate from chromosomal DNA and can replicate independently within the bacterial cell. In E. coli, bacterial conjugation is mediated by plasmids such as F plasmid. So conjugation is a horizontal gene transfer mechanism because conjugation enables bacteria to exchange DNA with neighboring cells. F plasmid, also known as F factor or fertility factor, is a circular DNA molecule ranging from 60 to 100 kilobase pairs in size, although variations can occur. Key components of F plasmid include ORI-T, origin of transfer. This is a specific DNA sequence where transfer process initiates during conjugation. ORI-V, origin of vegetative replication. This is a sequence where plasmid DNA will replicate in the recipient cell. Conjugative transfer genes or TRA genes. TRA genes encode proteins essential for the conjugation process. Conjugation occurs through specialized structures called conjugative pilus that connects the donor bacterium to recipient bacterium. TRA gene encodes a major structural protein of the conjugative pilus, that is pilin protein. TRA-B gene is involved in pilus assembly and stabilization. TRA-C contributes to the pilus structure and function. TRA-D gene is an essential component of type 4 secretion system, a machinery that spans the bacterial cell envelope and facilitates DNA transfer. TRA-I is a relaxase enzyme encoded by the F plasmid. Insertion elements, also known as transposons, are genetic elements that can move within a plasmid and cause mutations and altering the expression of nearby genes. F plasmid consists of a single copy of IS2 and two copies of IS3. Mechanism of conjugation. The donor bacterium carrying the F plasmid is referred to as F positive. The recipient bacterium in which the F plasmid is absent is referred to as F negative. F plasmid encode proteins that assemble a pilus. Pilus is a thin hair-like appendage on the surface of the donor cell. The pilus of the donor cell makes physical contact with the recipient bacterium. Recognition and specific binding between donor and recipient cells are mediated by surface proteins and other recognition factors. As we have already said, the F plasmid contains ORI-T sequence, origin of transfer, a specific DNA sequence where the transfer process initiates. Relaxase enzyme recognizes ORI-T and makes a specific single-stranded nick in the plasmid DNA at the site. Upon nicking, relaxase remains covalently attached to the 5' end of the nicked strand of plasmid DNA, forming a relaxosome complex. Relaxosome complex include other proteins that assist in DNA processing and transfer, such as coupling proteins that stabilize the interaction between the pilus and the recipient cell. The single-stranded DNA is then transferred through the pilus from the donor cell to the recipient cell. This transfer is powered by energy provided by ATP hydrolysis and guided by proteins within the type 4 secretion system. Now the donor cell and the recipient cell both contains single-stranded DNA of F plasmid. A complementary strand is then synthesized in both the donor and recipient cell. Thank you for watching.